Hi everybody, this is Ben Baldwin, and today I'm going to talk about testing and purity of essential oils. So first I'm going to cover, the importance of purity. Purity is an important characteristic in a good essential oil. It basically means that you know what's in there, you know what it's going to do. If the essential oil is pure, it's going to give you a therapeutic benefit you're looking for. If it's not pure, on the other hand, it may give less of a therapeutic benefit, no therapeutic benefit at all, or it could potentially even be harmful. So purity is pretty important, and testing plays a part in that purity. Now, what do we mean by purity? So let's go into a definition. Here we call the definition we're going to use is purity means that the essential oil is free from fillers, pesticides, solvents, additives, and is natural. It's not synthetic. Impure means that neither one of them has other stuff in there, or two, it's just not the way nature made it. And for sometimes, synthetically produced or adulterated essential oils may smell or look the same as natural or pure essential oils. So why do we need to test them? Well, we test them to ensure purity, because there's a lot of sophisticated tests that we can go through to find out whether an essential oil is pure, because we want that therapeutic benefit when we go to use it. Now here's an example of an essential oil produced in a lab. This is methyl salicylate, an essential oil found in wintergreen. It's a compound found in wintergreen essential oil, and it's also found in a lot of muscle rubs. This can be easily fabricated in a lab. Actually, it's less expensive sometimes to make compounds like this than it is to produce water, drinking water. So you take phenol, which is a strong acid, add carbon dioxide, which is poisonous, sodium hydroxide, very caustic, hydrosulfuric acid, which is very dangerous. You have to use a fume hood and all sorts of protective equipment to work with this. With methanol, which is poisonous, and you come up with methyl salicylate, which will also have trace amounts of the ingredients in the final solution. There's no way you can produce these things in a lab without having these really sometimes minuscule amounts of the trace solutions, but they're in there, and they make the essential oil impure and sometimes very dangerous to use. Even though they're widely used, the essential oils, remember, are not always used for therapeutic benefit. Sometimes they're produced for perfumes or for some other reason. So that's one reason why laboratory synthesis is not good for us. The molecule that's produced is exactly the same. When we're talking about the methyl salicylate, it's exactly the same, but whether there are trace amounts of the other additives, the ingredients that are also in that solution. That's the thing we're talking about. Now, nature produces these in a much different way. It uses, on a cellular level, the DNA instructions and assisted by proteins and the other parts of the cell that kind of read those instructions and put this molecule together. And you'll see as we talk, it comes up with sometimes a very different result. One of those differences is chirality. Now, sometimes the atoms in the molecule might be exactly the same, might be arranged in the same manner, but they're actually mirror image. Those produced by nature are quite often the case. Not always. Those produced by nature are, sometimes like a right-handed version of the molecule, whereas those produced in a lab are going to be 50-50. That's just, it's made in a solution, whereas nature makes it in a kind of a assembly line. So, and this is one problem with essential oils made in a laboratory, because the other orientation of the molecule may not be as therapeutic, and it might actually be harmful in some cases. Maybe cancel out the therapeutic benefit of the orientation of the molecule that we want. So this is one problem with making essential oils in a lab, is the chirality. And it's not going to give us as good as essential oils as before. Another problem with making essential oils synthetically in a lab is that you're isolating it. So nature, when nature produces essential oils, it's going to produce this constituent along with like 30 to 300 other compounds that are found in that essential oil, and in roughly the same ratio every time. Now, scientists have come along for years and they say, okay, this is the one that's actually giving the benefit. So they choose that one and they find out how to produce it in a lab. They produce it and it might produce some therapeutic benefit, but nature can do this a lot better by producing pure, not only the compound that we're looking at, but other assisting compounds along with it. Sometimes scientists regard all the other compounds as maybe junk, unnecessary, but it's been found over and over again that these other constituents in the essential oil actually help and benefit, and they work synergistically with the compound that we're looking for. It turns out that their junk was actually necessary, that nature was making those things for a reason. And having the right amount of each constituent provides a higher therapeutic benefit, which is why we're going to that essential oil in the first place. Some other reasons where that make an essential oil impure: adulteration, sometimes substitution. When an essential oil company, they don't have enough of this type of essential oil, they can find another essential oil that smells the same, tastes the same, it may have a lot of the same qualities, and they can switch those or spike their solution with something from another essential oil. It's kind of misleading, and it's not going to be exactly what you're looking for, and you may not get the same solution every time. You want to know that, and a good example of this is cassia and cinnamon. Whereas, if quantities of one essential oil are running low, sometimes it can be switched over because these both smell the same. They have very similar qualities, but they're not the same. Another way that essential oils can be adulterated is through the use of pesticides or other problems with the plant itself, impurities that the plant picks up. And so when that is, the essential oils are extracted from the plant, out come the impurities as well, and they go into the essential oil and make the essential oil impure. Additives and fillers are another way that sometimes essential oils become impure. When the producers of an essential oil, they may add some extra things into it to spike it, maybe wanting to improve its therapeutic benefit in a way, and they come up with something that nature didn't produce. And sometimes it dilutes it, or it makes it less expensive to produce, and sometimes it causes problems. But the fact is that you kind of get less of a therapeutic benefit than you wanted, and it's just another way that this thing can go wrong. Solvents is another way where these essential oils, to extract them, a cheap way to extract the essential oils from a plant is through the use of solvents. The problem with this is that some of the solvent actually ends up in the end product as well, also causing some impurities. So the way that we can find out whether any of these things have happened is through testing. That's what we're going to talk about now. There's lots of different tests. Some of them are very simple, some of them are very sophisticated, and we're going to go through a few of these tests. First is organoleptic, or sensory evaluation. It's basically just smelling, feeling the texture, tasting it if it's pure enough to taste, looking at it, the color, the flavor, the feel of it, the viscosity, how thick it is. Now these seem very simple and obvious tests, but they're the first starting point when testing an essential oil. Next is specific gravity. Does it sink or does it float? Specific gravity means how much mass is contained in a fixed volume of the solution. This is measured at a specific temperature, at a specific atmospheric pressure, and for example, the specific gravity of water at room temperature at sea level is 1 gram per milliliter. Oil is usually less than this, and so it floats. Not all essential oils do. Clove and wintergreen and cinnamon sink because they'll have a specific gravity that's going to make it. It's going to be more than the essential than water. This test merely points out that something may not be right with the solution. So you can do this test with your wintergreen or clove or cinnamon to see if it sinks or floats. It's a test to find out what its specific gravity is. And of course this will vary depending on the temperature or atmospheric pressure. Refractive index is another test that can be carried out. Now, as light passes through a substance, it slows down. The ratio of the speed of light as compared to a vacuum, like in space, and comparing that to the speed of light as it passes through a substance, like air, like glass, or a lens, is the refractive index. And this phenomena is what describes what happens like when light seems to bend as it passes through a glass of water, where it kind of distorts the image a little bit. And this ratio of how much it bends or how much it slows down is what we're measuring. And when you pass light through a substance, you can compare this refractive index to historical records of good samples, and thus determine whether there's something impure or something not right about the solution. So that's one way of detecting impurities. Next is optic rotation. Optic rotation merely means sending polarized light through a substance and measures how much that polarized light bends. Now, when the direction or angle changes as it passes through the substance, we can learn a lot about the molecular makeup of the substance. One of the important things we can learn here is the chirality, as we talked about earlier. That means the left hand or right hand version of the molecule can be detected through this test, and the polarized light will bend according to what the chirality of the solution is, and so nature curiously just produces that one chirality most of the time. It's not always the case, but it can tell us whether something is not right, or whether this solution is made by man or made by nature. So that's one test among many. The gas chromatography mass spectrometry test is probably the bread and butter of an essential oil scientist. This is the main test, most relied upon, and it gives the scientist so much information. It works basically by injecting the essential oil into an apparatus. The essential oil is vaporized and then propelled through a tube. And the essential oil compounds, the specific molecules in the essential oil, are basically segregated or separated based on their volatility, which means their ability to evaporate quickly. As these groups of compounds pass through in electromagnetic fields, the specific compounds are measured in terms of mass. So they're separated even further in terms of mass and quantity. So as they come through the mass spectrometry machine, they're then measured, and that's produced out on a graph. The heavier molecules are on this side of the graph, lighter molecules on that side of the graph, and the height of the point on the graph determines the quantity, or how much of that specific compound at that specific point of mass is in that solution. And these can get very, very detailed. You can run these over a longer period of time to get an even more detailed and precise test. You can see this sort of test can give scientists quite a lot of information. It's a simple test. A lot of impurities can be detected because they're going to show up on one of those spikes there, and the quantity and type of each molecule is also measured, so you know what this ratio is compared to another, like historical samples. This test can also detect the ratio, and the ratio is very important when it comes to the therapeutic value, because one, like lavender plants for instance, from one place will be different from a lavender plant from another place, although the molecules are going to be roughly the same depending on their species. The ratio may be different depending on the time of year, or the harvest method, or the soil, or the weather conditions that it's grown in. So this can also give us clues as to the therapeutic potency of the essential oil, which we'll talk about in another session. But what we're talking about here is whether it's pure. So this will tell us whether it contains some other molecule in there. When the molecule is discovered that's outside the ratio, for example, melaleuca. The terpenine for oil found in melaleuca is normally between 30 and 55%. So if it's outside those ratios, we know that something's wrong. This is not what's normally done by nature, and then we can determine this sample is impure. Maybe run some other tests to find out why. One other test that might be run is a Fourier transform infrared spectroscopy, FTIS. This is similar to the gas chromatography and similar to the optic rotation. It gives data similar to the gas chromatography or GCMS test. It works by projecting infrared light through the substance while it's rotating a mirror, and it can detect the characteristics of the bonds between the different atoms in the sample. It does this by measuring patterns in what infrared radiation is absorbed by the sample and what infrared radiation is allowed to pass through the sample. Now, after some complex computer analysis, this test can also detect the types of the molecules in there and their relative quantities. An interesting thing about this test is it also can detect the chirality, or the orientation of each molecule. That'll bring up lots of things like whether this sample is pure, or does it have any problems with it. This test can help substantiate the results of other tests, and it can help give scientists a bigger and better picture of what is in the sample and whether it's pure or not. So those are all the main tests. I just want to mention a few other tests that are very helpful and interesting to mention here. Carbon-14, we always hear about carbon-14 when it comes to dating ancient artifacts. So that's how it's usually used, and this is how it's used in testing essential oils as well. The theory is that different levels of carbon-14 an object has indicates when that object, or when the substances in that object were alive, essentially when plants were pulling carbon out of the atmosphere as opposed to carbon that's found naturally in the soil. Synthetically produced essential oil compounds use a carbon from sources that are sometimes very old or inorganic, and the test will illustrate this. So it's we're testing how old it is, and we can tell whether it's been made in a lab or whether it's been made by nature. The simple tests are done to determine whether whether a solution contains heavy metals or pesticides, and there's a wide range of those tests, but they're just simple, sometimes litmus paper type tests. And it's also important that an essential oil is tested before and after bottling for microbes. We call it TCP, or microbial testing, and they can then keep those samples for a long time and to see if any microbes are forming within the essential oil. So again, it's something to help us know whether the essential oil is pure, whether it's good, whether it's going to be therapeutic to us or not. So it's the testing that comes from this, all these different tests that give us the confidence to use an essential oil, that we know it is pure, it is going to be therapeutic, it ensures that it's safe. So pure means that it's safe. You also want to make sure your essential oil works. Pure is only half the battle. If your essential oil is pure, you're halfway there. The other half is therapeutic, and that is a different story for another time. But I hope you've enjoyed this video on essential oil purity and testing, and I hope you come and visit us and look at all of our other information on our website and our blog, our YouTube channel, and all that. Thank you.